Good morning students. In last video we were talking about the DNA fingerprint and we learned that the L.S. Jeffrey used a satellite DNA as a probe to make the DNA fingerprint. Okay, and this segment or this satellite DNA is also called VNTR, means variable number of tandem repeats, because this VNTR, this is the segment of DNA, part of satellite DNA, that the same segment, same sequence tandemly repeated many times. So this shows high degree of polymorphism, means the number of repeats or copy number varies from person to person, chromosome to chromosome. So first of all, to identify any person, we take the sample, we extract the DNA or isolate the DNA or chromosomes from a cell, any cell we can use, because all the cells of the body shows the same degree of variation. After isolation, what we do? The fragmentation of that isolated DNA. For fragmentation, we use the specific enzymes, that is called the restriction endonuclease. There are the different types of restriction enzymes. That why they are called restriction enzymes? Because they are specific to cut the DNA at a particular site. That site is called the restriction site for that particular enzyme. So there are the different restriction endonucleases enzymes that cut the DNA segments at particular site. This VNTR, now we'll talk about this VNTR. Suppose that this is the segment of VNTR. This is also bounded by the restriction site. Means both sides there is the restriction site. And so a specific restriction enzyme that cut that VNTR. So according to this VNTR, we uses the different restriction endonucleases that cut the VNTRs in different fragments. So after now this is the fragmentation or digestion of the isolated DNA. Now after that we separate the fragments. Separation of fragments. Now what is the technique? That how we separate the fragments of the VNTR or fragments of the DNA? So there is a technique that is called the gel electrophoresis technique that we will study in the biotechnology topic. In gel electrophoresis there is an electric field, cathode and anode. We know that the DNA is the negatively charged molecule. So at the cathode end, we use the DNA and force the DNA to move towards the anode. And there is the matrix that made of agarose gel that acts as the sieving effect. Sieving effect. So now the fragments of DNA move towards the anode. Small fragments move faster. Larger fragments move slower, and the different fragments in this agarose gel that acts as the sieving effect, the different fragments will be separated. So this is called the separation of the fragments. So VNTRs are now separated. After separation of this VNTR, now after separation what we do? We can do the we can make the single stranded DNA. How? So we can now we treat these fragments by the alkaline chemical that breaks the hydrogen bond between the double stranded DNA. So the strands will be separated, means it will produce the single stranded DNA. So by the use of these alkaline chemicals, we make or we produce the single stranded DNA. Means we know that the DNA is the double stranded. So this alkaline chemical break the hydrogen bond between the two strands, and so the strands will be separated. After this production of single stranded DNA, then another technique is called the Southern blotting technique that developed by the Southern. This is the name of a scientist. In Southern blotting technique, what we do? We transfer this fragmented and the separated DNA, single stranded DNA. We transfer on a nylon sheet or nitrocellulose membrane sheet that is placed over a gel. There is also gel. That gel acts as the blotting. So we transfer that single stranded DNA on nitrocellulose membrane or nylon sheet that is placed over a gel. This is called the southern blotting, developed by the scientist, name is the southern. After this southern blotting, now we put out that the nylon sheet or the nitrocellulose membrane and immersed in a bath. We immerse that membrane in a bath. That bath contain, now very important thing, that bath contain DNA probes. Question is, what is DNA probes? So DNA probes are actually synthetic DNA of known sequences. So these are the segments of DNA made by the scientists, that is the synthetic DNA having the known sequence of nitrogen base, and that sequences are complementary to the VNTR. VNTR is the single stranded, and now the probe is also single stranded, and the base sequence of the probe is complementary to the VNTR. Means this DNA probes used to identify a particular VNTR. And second thing, this synthetic DNA or DNA probes are radio labeled, means they are made radioactive for identification. So this nitrocellulose membrane is immersed in a bath containing DNA probes. And what I said, what we learned, what is this DNA probes? The DNA probes are the synthetic DNA of the known nitrogen bases or sequences which is complementary to a particular VNTR, that is single stranded VNTR. So when we immerse the membrane in the DNA probes, this synthetic DNA now attached with the VNTR, identify the attached with the, and the double stranded DNA will be produced. That is called the hybridization of the DNA. And the probes were what? It will be the radio labeled, that helps in the identification. So now after hybridization, this hybridized VNTRs, this is hybridized DNA. Now it is undergoes now the another process that is called the autoradiography. Means we expose to the X-ray and take a X-ray film. This X-ray films gives the different bands, and that different bands are actually the fingerprint of the VNTR. That is the different bands that it shows. That of different bands, there will be the different length of that band. It depends upon the number of number of repeated units of that particular VNTR. So different bands will come. That is the DNA fingerprint. So this is the DNA. Now this fingerprint is now used to compare the fingerprint of another person. Suppose there is a crime, a place, and at the crime scene we have collected a sample, hair follicle or skin cell or semen or blood, whatever, anything, and we isolated the DNA and we make a fingerprint. Now we have, suppose, the two suspects, suspect A and suspect B, suspect Ram and suspect Syam. Suppose they are the suspects, that the police have suspicion that they are the person may be committed the crime. So what we do, we also take the sample of that two person, and by the same process we make the fingerprint of this two person also, and then we compare the fingerprint of the crime scene. So this is the, and suppose there is the two suspect, and one suspect, the fingerprint of one suspect matched with the fingerprint of the crime scene, means that person committed the crime. So this is the process that we involve in the DNA fingerprinting that we studied. So suppose we have any crime or any paternity dispute also, in that condition also this fingerprint can be used to identify a particular parent in paternity dispute. In forensic science also it can be used to identify a particular person or any criminal who have committed any crime. So this is the process of DNA fingerprint. Suppose there is any case, any crime case we take the sample, cell collect the sample from that crime scene, we make a fingerprint. Suppose we have the two suspects A and B, their also fingerprint we prepare, and then we compare the fingerprint of A and B with the fingerprint of the crime scene, and we can compare, we can find that who have committed the crime. So in this way we can identify the criminal also. So this is the process of fingerprint. Thank you.